This video is going to take you through the first Taxonomy Lab. We're doing the more primitive organisms, and so all of the prokaryotes, which is the bacteria and the archaeans, happen today. We're also going to talk about protists and fungi. Next week we're doing plants, and then the last week we're doing animals. And so we're staying in taxonomy for the whole last chunk of the lab, and lecture for that matter. This is a lot of information that a lot of students sort of resent having to learn. And I understand why. Taxonomy has been in flux since the 1990s. Every textbook that we use provides a different classification system, which means lecture and lab don't agree with each other. Your lecture textbook doesn't agree with your lab textbook. If you were to search on Wikipedia, it wouldn't agree with anything else. And so there's a bunch of different Latin names that are flying around. What I would recommend that you do to help you study this information is make some flashcards. Now, another thing that can really help you out as we go through here, let me find the right page. There is a table that starts on page 288 in the lab manual. It's number 7. It's one of those post-lab questions, but filling in this table can really do a lot to help you understand the organization and the taxa that are occurring with each of the different specimens that we're going to be going over. So I'm going to recommend that you fill this table in as we're doing the slides and learning the information, and I'll tell you if you have to flip to other pages to fill in some other stuff, but I don't know, put a little post-it note or something on page 288 so that you can keep flipping back to that table and fill that table in as we go. So to get us started in this chapter, I threw in some words a second ago, taxonomy and taxa, so you need to know what those are. So your definition for taxonomy is up here at the top. Basically what it means is if you were to find a new species, you would have to put it into a certain group based on its characteristics. So let's say you found something that was furry and it had bones, like it was this weird picture that you're seeing right here. Well, you would put it in the domain Eukarya, which your lab manual doesn't use that domain, but most textbooks do. So I'm going to stick with that domain. You would put it into that domain because it had eukaryotic cells, which it's hard to tell from this picture, but trust me, it has eukaryotic cells. You would put it into the kingdom Animalia because it's multicellular. It's pretty clearly some kind of animal. It's not a plant. It's not a fungus. It's not a protist. And so kingdom Animalia. You'd put it into the Phylum Chordata because some of the features that it has are a backbone with a spinal cord that runs through that backbone. And so that's what goes in the Phylum Chordata. Because you notice it had hair, it would go into the class Mammalia. If you were to watch this critter long enough, you would notice it eats things like mice and voles and rabbits and hares. And so that means it's a carnivore, so it would go into the order Carnivora. It's kind of dog-like, and so it would go into the family Canidae, which is where all the dog-like things go. But it's not like a wolf. It's too small for that. Instead, you would put it into the genus Vulpes, which is where foxes go, and then Vulpes is just the red fox. So that's how these taxa work. You look at the characteristics that it has, and then you put it into certain groups. Now, the groups up at the top of this picture are the most inclusive, and what that means is there's just 3 domains, and every living thing on the planet is put into one of those 3 domains, based on the characteristics of the cells that make up the organism. From there, things start to get a little bit more specific. The number of kingdoms varies wildly depending on what textbook you use, but there's somewhere between 5 and 20, more than likely. There's a bunch more phyla, there's a bunch more classes, and there's millions of species of things that are on the planet. So species is the least inclusive or most specific taxa. A taxon is a level in this. So a species is a taxon, a genus is a taxon. The plural for taxon is taxa. So each one of these is a taxonomic layer. This is what's known as the taxonomic hierarchy. Your lab manual gives you a couple of stupid memory tricks that can help you remember them, although your lab manual, again, does not use domain, but most textbooks do. So they give you mnemonics that just include kingdom through species. King, what did they give you? King Philip called out for good ships. Kids playing chicken on freeways get squished, but remembering a mnemonic can help you remember the order in which these occur. The reason why that's helpful is because each thing is a group of the things below. So a genus is a group of related species. A family is a group of related genera. An order is a group of related families. A class is a group of orders. A phylum is a group of classes. A kingdom is a group of phyla. And so that's why the mnemonic can really help you remember what's going on there. All right, after taxonomy, we start to talk about some of the important people related to that. Your lab manual goes a little bit into Aristotle. One of the more important people related to this, his name was Carolus Linnaeus. He traveled over a lot of different places and he named a bunch of different species. And when he named them in Latin, and he named them based on which taxa they belonged in. The genus and the species is what he used to make up the scientific name, which is what's known as the binomial name, or the process of using the binomial name is called binomial nomenclature. So we are Homo sapiens. An oak tree is Quercus virginianus. That fox from a second ago was Vulpes vulpes. One of the things that we have started to use to help us out with taxonomy in recent years is a field known as phylogenetics. So this uses genetics to group things into sort of family groups, and the families are based on evolutionary relatedness. Now, we talked a little bit about DNA similarities when we were talking about evolution. So when we look at organisms' DNA, the organisms that have more similar DNA, they fall out into groups. And then we can make this thing that is called a phylogenetic tree, which is sort of a family tree for different things. So I decided to make this current and relevant. This is the phylogenetic tree for COVID-19. COVID-19 is the one that's in red right here. So what you can see by looking at this is it's most related to SARS, which was, 2003 or something was when SARS came to be. It's related to some bat coronaviruses, which is why a lot of people think that this particular virus originated from a wet market where they were selling bats for human consumption. There's more bat coronaviruses that are a little bit more distantly related, but they shared a common ancestor back this far. And so what they're doing is they're grouping things based on that DNA relationship between them. Now, phylogenetics has really been practiced since well before we had DNA available, but when we did it back then, we were looking at characteristics that were visible to the naked eye. We wouldn't have been able to do that with viruses back then. So now that we can use DNA relationships, we've really been able to make more accurate phylogenetic trees. Okay, let's see. We're starting with Kingdom Archaea. Once again, I'm going to recommend that you're starting on that question number 7 on page 288. Although then again, it's not in the same order of things that we're kind of going with right at this particular time. It looks like they actually did not put, I'm looking at the table right now, in its entirety, and I actually don't see Kingdom Archaea represented on here at all, looking at the species they started. It looks like they started with the regular bacteria, so I guess never mind, go back to page 276. This is really a domain based on rRNA sequences, and again, most textbooks do put it into a domain. They do mention in the lab manual they're using a very specific Library of Science classification system. It's using old evidence, and so that's why I prefer to call this a domain, because that's what the newer evidence tells us that it is. So, domain Archaea consists of prokaryotic organisms. Remember, prokaryotes have simple, small cells. There's no nucleus. There's no other membrane-bound organelles like roughy R, smoothie R, Golgi apparatus. There's no mitochondria. There's no chloroplasts in the kingdom or domain Archaea. To make life a little bit more simple for us, what we're also going to say about the Archaeans is they tend to live in extreme environments, places that are really hot or really salty or really acidic or really basic. Places where we did not think life could exist. Turns out these guys have been living there all of this time. This is a pink lake in Australia, and one of the reasons that it ends up being this vibrant pink color sometimes, it's not always pink, but when it is, it's because there's a bloom of a specific halophile species that is an Archaean. So halophiles are things that like salt. Usually, so phile means you like something, and then whatever comes before it tells you what you like. So thermophiles like hot, halophiles like salt, acidophiles like acid, alkalophiles like basic or alkaline environments. Next, kingdom bacteria, again, most evidence says this should really be a domain at this point. And so now for sure, we're flipping back to page 288. Staph aureus, Strep pyogenes, Bacillus anthracis, the one that currently just says Proteobacteria and the one that currently just says photosynthetic bacteria, those are all in the same kingdom, which really should be a domain at this point. But they're in the, what you're going to put down as kingdom bacteria for lab. Everything in this kingdom is prokaryotic, just like the Archaeans were. But again, to make it more easy to understand, we're going to say these aren't extremophiles. They live everywhere. Now, after we have filled in kingdom bacteria for all those organisms on the table, we're going to flip back because we can use the shape of an organism and the arrangement of the cells to help us identify a species of bacteria. The shapes that are present in most bacterial cells are coccus, bacillus, and spiral-shaped. Coccus means spherical or ball-shaped. Bacillus means they look sort of like a dash or a minus sign, so they're rod-shaped. And spirillum means they're corkscrew-shaped or they spiral. The arrangement. And they just put staph and strept, but the full prefix is staphylo and strepto. So staphylo means they occur in clusters. So, for example, Staphylococcus aureus, it's staphylo because they're arranged in clusters, and it's coccus because they are ball-shaped. And so Staphylococcus aureus is called that because it occurs as little ball-shaped cells that cluster together. Strepto occurs in chains. Streptococcus would be a chain of coccus organisms. There can also be streptobacilli. Bacilli don't really do staphylo arrangements though, but here's your streptococcus arrangement, and Streptococcus pyogenes is the species that causes strep throat, which again, we'll talk about that a little bit more later on. So make some flashcards of those shapes and arrangements because those are going to help you recognize the bacteria that we're about to start looking at. All right. Phylum Firmicutes. Some people elevate this up to a kingdom, but we're going to call it a phylum in this level. Everything in this phylum is gram-positive. Now, a gram stain is what's known as a differential stain, and it's called that because it stains different organisms different colors based on the anatomy of the cell wall of that organism. Everything that is gram-positive ends up looking purple after a gram stain, and so all of these are all gram-positive. So, flip back to the table, page 288. Staphylococcus aureus, Streptococcus pyogenes, and Bacillus anthracis, they are all in the same phylum. So for those three species, you're going to put them in the phylum Firmicutes. The characteristic that you're going to write down for that phylum, they're all gram-positive. All right. They have given you different pictures from what I have. Theirs are sort of like weird animated pictures, but here's how you would recognize each one of these individual organisms. It goes back to shape and arrangement. So notice that in this picture, all of the cells look like little teeny tiny balls. That means coccus. And they are arranged as clusters. There's a small cluster, here's a bigger cluster, and cluster is staphylo. So this is Staphylococcus aureus. This one occurs in chains, and we're still little ball shapes, and so this is Streptococcus pyogenes. This one is not ball-shaped, it's rod-shaped, so that's a bacillus, so this is Bacillus anthracis. So Staph aureus, Strep pyogenes, Bacillus anthracis. Staph aureus, that's an organism that if you've ever heard of a staph infection, Staph aureus is the organism that causes a staph infection. It can be like a wound that you got that didn't heal. It can also sometimes produce a toxin that can do some really horrible things to you, but we won't really talk about that here. Strep pyogenes causes strep throat or strep pharyngitis. Most of you have probably had that at some point in your life, so you know what that one's like. Hopefully none of you have had anthrax though. So anthrax causes respiratory distress that can lead to death, and so anthrax is not one that you really want to play around with. All right. Next, in the table, they have already put Phylum Proteobacteria in there. This is still kingdom bacteria, so that's what you would put in the kingdom place. For the characteristic of this phylum, they're gram-negative, not gram-positive. Going back to that gram stain, these guys stain pink instead of purple. So anything that ends up being pink under the microscope in the bacterial world would be considered gram-negative. Your example species for this one, so the organism that you're going to write into the line is Escherichia coli, which is more commonly just called E. coli. E. coli can cause anything from diarrhea. It is the most common cause of urinary tract infections, which is what I've put up here, but that's not what they said in the lab manual. They said, well, no, they did say it can cause urinary tract infections. Food poisoning, traveler's diarrhea, and even death in extreme cases is what your lab manual says for that, but this is where most people tend to get their E. coli. All right, back to the table. They put photosynthetic bacteria, formerly called blue-green algae. The name is nostoc, so this is what you would put in the organism group. Kingdom, still bacteria. phylum is Cyanobacteria. So these guys don't have to be gram-positive or negative because they make their own pigment. They look bluey-green under the microscope even when you don't stain them. And so if you're looking at bacteria and they happen to be green, then you know it has to be Cyanobacteria. This is where that stupid game Among Us can help people out because apparently one of the creatures is called cyan. Well, cyan has always meant blue-green, and so blue-green. There you go. All right. Next, we're moving to a completely different group. This is the domain Eukarya. This used to be called Kingdom Protista, but now it's the Kingdom Protozoa because some of the other ones, the algae, have been put into different kingdoms at this point. These are the animal-like protists. Your characteristic, let's see, this is going to go starting from Euglena and going all the way down to where it just says Apicomplexa in the phylum group underneath Paramecium. All of those, so it's, let's see, Euglena, Trypanosoma, 2 blank ones, Paramecium, the next blank one. Those are all in this kingdom Protozoa. So these are eukaryotic. They have cells that have a nucleus. They are bigger cells. So this one right here, you can see the nucleus really easily in this cell. This is not a fantastic picture of it, but there's the nucleus for the cell that's inside of one of our red blood cells. Both of these happen to be human parasites. Not all protozoans are parasites. We tend to only be concerned with the ones that are. The ones that aren't, who cares? They don't hurt people, so we don't care about them all that much. But they did go ahead and discuss different kinds of relationships that can exist. And so at this point, we'll flip back forward to look at these definitions that are on page 280 in the lab manual. So parasitic, commensal, and mutualistic. These are all what are known as symbiotic relationships. Symbiotic means you have 2 different organisms that are living together. Then each specific term tells you how they're interacting with each other. So parasites, one organism wins, the other one loses. In this case, the parasite wins because they have a food source. The person that they're living in or on, they're losing because they have a parasite that's causing an infection and they're going to have symptoms as a result. Commensal, still 2 species living together. One wins, one doesn't do anything. It doesn't win, it doesn't lose. And then our last option is mutualistic. Now, mutualistic is very often confused with just the term symbiosis. Mutualistic means 2 species are living together and everybody wins in the process. So what I want to make sure you understand is all of these are symbiotic. Mutualistic is just the one that means everybody wins. So symbiotic doesn't mean everybody always wins because it could mean only one wins and it could mean one wins, one loses. And so just make sure you understand the distinction between those words. All right. Next up. One of the things that we can use to classify our different protozoans is how they move around. They can either have flagella, like this little guy right here. They can have cilia, like this middle one, or they can have pseudopodia. If they have flagella, they tend to swim around fairly easily. They use that sort of like a propeller. So this one happens to be Euglena, and I know that because it has a flagellum. Cilia, they tend to do more of a gliding movement as a result of the cilia, or they move other things around with their cilia. And so you'll see these little guys moving around, but you will very often not actually see the cilia because they're so small, they're really hard to see with a microscope. So if you see something moving, but you can't see why it's moving, it's probably cilia. Pseudopodia is the one that's hardest to sort of explain. What happens is the cell sort of smushes its cytoplasm out into this little projection. That projection is a pseudopod. So that's a pseudopod. That's a pseudopod that's getting smaller. There's a pseudopod that's getting bigger. Pseudopodia is plural for pseudopod. So if you see something that's sort of oozing around, like, this is going to really age myself, but if you guys remember the movie The Blob, they move like the blob. But they don't have cilia. They're just oozing around. So flagella, cilia, or pseudopodia. That's how we're going to classify our organisms now. All right. So flipping back. Euglena. We've already said it's a protozoan, so kingdom Protozoa. It's in the phylum Euglenozoa. This is one of the times where, yay, the name actually kind of goes together. The Euglena organisms can be photosynthetic if the water is nice and clear. If the water quality starts to get poor, though, they will actually kick their chloroplasts out and then become eaters at that point. So your characteristics for this particular phylum is this top one And for Euglena specifically, it can be photosynthetic. Now, for Trypanosoma, same kingdom, Protozoa, same phylum, Euglenozoa, but its characteristic is that not only does it have flagella, it's a parasite. This one is a parasite of human beings. What will happen, and it only occurs in Africa, sub-Saharan Africa. There's a fly, a biting fly called the tsetse fly. The tsetse fly bites a person and spits a little bit into the person. When they do that, they spit some of the Trypanosoma into the person. That leads to African sleeping sickness, which is called that because since they destroy your red blood cells, you don't transport oxygen very well, so you get very tired, and then you fall asleep and you never wake up if it goes to its poor conclusion. All right, next up, you have Phylum Amoebozoa already written down. The name of the organism is Amoeboproteus. This is still in the kingdom Protozoa. Your characteristic for this one is they move with pseudopodia. So the pseudopodia is actually this extension. They have actually, this particular amoeba has trapped a paramecium and is going to start digesting the paramecium. The paramecium in this image realizes he's doomed and he's just swimming around in a little vacuole inside of the amoeba at this point. So that's the Amoebozoan. Next, Metamonada is what you have next. So this is the one that says eukaryotic, lack mitochondria, have 4 flagella. So in the phylum box before that, you're writing Metamonada. In the kingdom box, this is still Protozoa. Your organism is Trichomonas vaginalis. So Trichomonas is an organism that causes trichomoniasis, which is a relatively common STD. It gets treated with antimicrobials that are kind of like antibiotics, but not, because this is not a bacterial infection. So penicillin, for example, would not work on this species, but there are treatments for this. Unfortunately, this one is also becoming resistant to treatment, just like the antibiotic resistance that we talked about with bacteria in an evolution chapter, though. So if you've ever had trichomoniasis, this is what's traipsing around in your genitals. Next, in the table, you have paramecium already written down. Paramecium is in the Protozoa. Well, actually, no, they're now calling it a different kingdom. Sorry. I'm not used to this kingdom. This one is a new one for me. I've never heard anybody else use this one before. So they're calling it the kingdom Chromista. So I'm sorry that I told you something different a little bit earlier. Kingdom Chromista is called that because of the chrome means color. They have their own color. They are, the paramecium that we're going to be talking about is in the Phylum Ciliophora. This is our group that has cilia to move around. So this is our paramecium swimming around using his cilia. I have never seen another source say that a paramecium is photosynthetic and has chlorophyll. I'm very iffy on this whole kingdom Chromista thing with the paramecium, but I'm going with what your lab manual says, and so this is what it says for the paramecium. All right. Phylum Apicomplexa. They want this one, I guess, to be in the Chromista kingdom as well. So once again, sorry, this used to be considered a protozoan, but change where I had told you to put kingdom protozoan to kingdom Chromista, this new one. Apicomplexans are called that because they have a complex life cycle that we don't, in a lot of cases, we don't understand all of the stages. Now, malaria is one of the most studied protists that are out there, and we know its life cycle. It has to spend part of its life cycle in our red blood cells. It has to continue part of its life cycle in the mosquito, in the salivary glands of the mosquito. It's got a very complex set of pathways that it has to take in order to survive. Your characteristic for this phylum is the top sentence that I've given you right there. Again, this should be kingdom Chromista at this point, but the organism that we're talking about here is Plasmodium vivax. This is just one species of this parasite that would be this little sort of, again, cyan cell that's moving into a red blood cell. It continues part of its life cycle in the red blood cell as the sporozoite, and then it leaves and it can go off to the liver and go off to other things. So again, it doesn't even just infect one cell in a human. It infects multiple organs in humans as well. And it does need that mosquito as a vector. All right. Still in that same group, same kingdom, Chromista, we have the Phylum Ochrophyta, which is again a brand new phylum that I have never heard of before, but whatevs. Phyta means plant-like, and so this is a group of protists that are very plant-like. And I think most of you, if you were to see kelp, you would probably call kelp a plant. Seaweed, same thing. However, there are also unicellular examples of these guys. And so let's see, you have diatoms written down, and fucus and kelp. All of these, kingdom Chromista. All of them, Phylum Ochrophyta. So they're all going to have these same sort of characteristics. They have chlorophyll, but they don't use the same chlorophyll that plants use. Plants use A and B. This particular group uses a brand new one called chlorophyll C. Diatoms you could only see if you have a microscope because they're single-celled. They are, if you have OCD, diatoms should make you really happy. They've always made me happy, and I do have OCD. They're so beautiful to look at under the microscope. They come like in every shape, and because they have a glass shell, they're prismatic and they reflect light, and it's just beautiful to look at them under the microscope. If you have ever used diatomaceous earth as like an insecticide or as a pool filter or anything like that, you're using the dead skeletons of diatoms. Fucus is just seaweed. If you have ever been to the beach and stepped on this slimy, gross stuff, then that's probably fucus. And then kelp is what makes kelp forests. We usually, the closest ones to us are actually off by California, like in Monterey Bay, which is kind of by San Francisco. They get kelp forests out there. It's like giant seaweed, like where seaweed is definitely visible to the naked eye. Kelp is hundreds of feet tall. It goes from the bottom of the ocean all the way to the top, and they create this whole ecosystem that lives in the kelp forest. All right. Next, switching kingdoms completely, we are going to the kingdom fungi. Everything from penicillium on down to mushroom, which is at the bottom of this table, they're all in the kingdom fungi. Everything in this kingdom is eukaryotic, so again, complex cells. They can be unicellular or multicellular. They do tend to grow in these little threads or filaments that you very often need a microscope in order to be able to see. But one of the other hallmark features of the fungi is they are decomposers. They can't eat something like we can. Like they don't have a mouth to put food in. Instead, they release their enzymes outside of the body and digest outside. And then once the nutrients have been broken down into small enough molecules, then they'll just absorb it through their body. And so they work differently. Their cells are sort of a mix between plant and animal cells. They don't get chloroplasts like plants do, but they do get a cell wall. Their cell wall, however, is made with chitin, is how you say that word, so pronounce it with a K sound, chitin, instead of cellulose, which is what plants use. All right, I'm trying to make sure I haven't skipped anything in all of these other pages for you guys. All right, so far, so good. Okay, let's flip back to page 284 really quick. I was so concerned with making sure we were filling out our table that I did skip something. It said, look at the fucus specimen in the lab room and draw what you see in the following space. So they just have in a jar some preserved seaweed. So this is what you should doodle. Now, one of the things to notice is it's plant-like. It's greenish. It's really called a brown algae, but whatevs. It's got these little air bubbles in it that help it stay towards the top of the water. Think sort of like a swim bladder, but for a plant-like thing instead of for a fish. And it's almost always sort of vaguely slimy. So that's what you should doodle in the space that they have given you on page 284. This is for your fucus. They've given you a little picture of diatoms, and then there's some, it looks like kelp that's washed up on a beach someplace where there's a dude standing next to it. So this is the only one they wanted you to draw there. All right. We already did that slide, so we're okay there. Okay, so back to the table, to penicillium. Penicillium, again, kingdom fungi. It's in the Phylum Ascomycota. So is Candida albicans, and so is Saccharomyces cerevisiae. So these are all in the same kingdom and phylum, these three organisms. Penicillium chrysogenum is the species of fungus that makes the antibiotic penicillin. This is what penicillin looks like when you grow it on an agar dish. They have given you a different picture of penicillium, and the picture that they gave you, I have always remembered microscopic penicillium because to me it looks sort of like skeletal hands reaching out. And so, I don't know, doodle a picture of penicillium with the little skeletal hands reaching out instead of this one. This is macroscopic version. They gave you the picture of the microscopic version. Candida albicans. This is one version of it. They gave you a stained version. Mine is unstained. But this one, I don't know. I've always kind of thought just I don't even know how to describe what I thought. Sort of vaguely cherry blossomish is how I've always kind of thought Candida albicans looks. I've heard some people say like fireworks, which I don't see, but if you see fireworks when you look at this one, awesome. Candida albicans is the species that causes yeast infections in people. Yeast infections can occur in all sexes and all ages. It is not just a woman disease. It can occur in the vagina, in the rectum, in the mouth. It can occur on feet. It can occur basically anywhere that there is a moist, wet environment. Last, Saccharomyces cerevisiae. This is the species that if you've ever gone to the store to buy yeast because you were going to make bread, this is the species that's in that little pouch. So all those little brown grains that make your house, I've always thought baking bread smells amazing, but some people hate that smell. But whatever that smell is, that's the fermentation of this yeast so that you can actually make your bread, or if you're a home brewer, your beer or wine or whatever. So I've seen them on practicals literally just throw out a packet of that yeast and ask you what it is. So it's Saccharomyces cerevisiae. It's an Ascomycota, or an Ascomycete is the way you would actually say that. And then it's a fungus. All right, Zygomycota. It just says in the table form zygospores. So before that box, you would write Phylum Zygomycota. Still a kingdom fungi. The organism for this one is Rhizopus, is usually just what we call it. Rhizopus is common black bread mold. So this is what it looks like in macro form, and then this is what it looks like if you look at it under the microscope. So this one, I've always thought it kind of looks like Bob Ross. So this is his head, this is his lovely little afro, and so little Bob Ross is under the microscope. Last, or wait, nope, they wanted you to draw a Rhizopus, sorry. So flip back to page 286. Ooh, and they wanted you to draw a Saccharomyces. So look at the Saccharomyces cerevisiae and the Rhizopus specimens in the lab room and draw what you see. Again, in the lab room, they literally throw that packet out there. So put yeast, rapid rise, or bread yeast for that. But this is what it looks like under the microscope. So you can pause the video and doodle either one of those for Saccharomyces. It is a unicellular yeast, unlike these other two, which were multicellular. Then, for Rhizopus, I recommend you draw the Bob Ross for Rhizopus, because this is usually what they show you a picture of. And it's usually pink because they will have stained it. This one looks a little bit different because the picture's not stained. But there is your little Bob Ross Rhizopus. Poor Bob Ross. I wonder if he would be sad if he knew I thought he looked like bread mold. Phylum Basidiumycota. So you have in the table on page 289 at this point, mushroom. Kingdom fungi, Phylum Basidiumycota. Oh, that should be an O. It's Basidiomycota. Sorry, I have a typo. I'm going to blame autocorrect, even though it probably didn't do it. Your characteristic is it's a club mushroom. The club is like if you've ever eaten a mushroom, it's what you ate. So that's a club, that's a club, that's a club. There's little clubs that are starting to come up. If you flip it over, it has gills underneath it. But your typical mushroom shape that you have a relationship with, it's in the Basidiomycota grouping, and the club is their reproductive structure. Let's see. So agaricus, that's just portobello mushroom or whatever. It says to look at the mushroom specimens in the lab room and draw what you see in the following space on page 287. Just draw a random mushroomy type thing and you should be good to go for agaricus. They usually just have like a couple of little button mushrooms in a jar. The other thing that I usually like to say in relation to mushrooms, if you are ever in a situation where you're really, really hungry and you're starting to think, well, I'm going to eat anything and this mushroom is here, do not eat a mushroom unless you know for a fact that it is not toxic. Because if you eat a toxic mushroom, it is a very unpleasant death. Most of them have toxins that destroy your liver. You'll become jaundiced and painful as waste starts to accumulate in your body, and then you die several days later. So just don't play around with mushrooms. Some of them you will trip balls until the day of your death, but most of them it's just a really painful death. And so just don't mess around with mushrooms unless you specifically know exactly what they are and what chemicals they might produce. All right. After that, flipping back to page 287, we start out with some questions. Once again, I'm just going to tell you, pause the video and get what you need. So there's 1 through 5. Here's the table for question number 6. Question 7 is that table that I've been telling you to fill out as we've been going through here. Number 8 is matching. Pause it and get what you need. Number 9, they give you pictures of all the organisms, and they are the exact pictures from your lab manual, and then they ask you questions about each one of those. And so here are the answers to the questions A through I. Again, pause the video, get what you need. For critical thinking, if two species of bacteria closely resemble each other, are they closely related? Why or why not? So if they are resembling each other, that means they have the same shape or maybe the same Graham reaction, and no, they don't have to be related to each other at all. Like E. coli is a rod shape, and so is, pardon me, the Bacillus anthracis that causes anthrax. They aren't related to each other at all, so no, they don't have to be related to each other because shape does not dictate relationship. Number 2, if a microbiologist were unable to culture archaeans from an environmental sample, should they conclude that there are no archaeans growing there? Why or why not? So when we culture something in a lab, it means that we are trying to grow it in either a broth or on a sample or on an agar plate. When we make broths or agar plates, we put specific nutrients into those and we try to mimic the growing conditions for certain species. Archaeans are very, very difficult to grow in a lab because they need such specific chemicals and nutrients and temperatures and salt conditions that it's just practically impossible for us to make broths or agar that they will grow on. It doesn't mean they're not there. It means that we just couldn't mimic those conditions very well. We don't have incubators that reach up to the temperatures that some of those things grow in, or reach down to some of those temperatures that those things grow in. Number 3, why do microbiologists study archaea when there are plenty of other species that can cause disease in humans? So, as a scientist, I think, well, just because all life matters. Like, just because something matters more to humans doesn't mean we shouldn't study something that doesn't matter to humans. If we're going to go to all the expense of sending people to the moon, shouldn't we study the things that are here on this planet first? It has always kind of been my thought. But if you need something more practical, well, anytime we find a new species, there's a potential that it can produce an antimicrobial chemical that we could use as an antibiotic to treat other bacterial infections. Especially when it comes to unicellular things like archaeans, they are competing with bacteria and they will very often carry out chemical warfare against bacteria. So there's probably chemicals that they're making that can help us in our fight against pathogens. We sometimes discover that they make drugs that can help us in other ways, like anti-cancer drugs. And so studying them can bring us new medicines, new foods, new processes. Like maybe some of them are better at photosynthesis, and so we can make food faster by using them. We don't know until we study them. Number 4, early classification of fungi put them in the plant kingdom. What characteristics of I think is what that's supposed to say, fungi and plants share? How are they different? Well, they can both be multicellular. They are both made out of eukaryotic cells. They do both have cell walls. However, the cell wall is different. The mode of nutrition is different. Fungi can also be unicellular. So there's plenty of differences as well. So they really should be 2 separate kingdoms. Even based on DNA evidence, they should be 2 separate groups. Number 5, how is global warming predicted to affect organisms of the kingdoms in this exercise? It's impossible to tell for most of these because, like I say, all species are affected differently. The ones that like warmer, more acidic environments, probably going to do better because climate change is making places warmer and it's acidifying the ocean. Places or organisms that like it to be more basic, they are going to die out. Places or organisms, that like it cooler, they are going to die out. And so some are going to win, some are going to lose. That's how that's going to go every time they ask that question. That concludes this chapter. This is a lot of information, but again, flashcards can really help you out with that. And then studying that table that we filled in, that should really help you out with that as well. From here, you're going to take the post-lab quiz. Good luck.